Think about the last time you went completely blank. Not tired, not distracted, blank. The kind of blank where a thought was right there, fully formed, accessible, yours. And then something happened. A phone call came in with bad news. Your boss walked into the room with a look on their face you recognized. An email landed in your inbox with a subject line that made your stomach drop. And the thought you were holding vanished. Not misplaced, vanished. And in its place was something that felt like static, a kind of gray electrical hum where your cognition used to be. Think about the last time you went completely blank. You have almost certainly described this experience as being overwhelmed or panicked or stressed. What you have probably never done is describe it in the language it actually deserves. A forced rerouting of prefrontal cortical executive function triggered by an amygdala-initiated hormonal cascade that physically suppresses synaptic activity in your brain's most metabolically expensive tissue. That is what happened. That is what is always happening when you go blank under pressure. And once you understand the mechanism, not in vague pop science terms, but in the actual thermodynamic and molecular detail of what is occurring inside your skull, you will never experience that blankness the same way again. Because it is not a character flaw, it is not a weakness, it is not a failure of intelligence or willpower or preparation. It is a piece of ancient engineering doing exactly what it was designed to do. And that engineering was designed for a world that no longer exists. Here is what nobody tells you about your brain. You do not have one. You have at least two, and they are not partners. They are competitors. They share the same skull, the same blood supply, the same evolutionary history, and they have been arguing with each other for approximately 200 million years about who should be in charge. And the rule they operate by is simple and brutal. Whoever gets the glucose wins. The first brain, the one you think of when you think of thinking, is the prefrontal cortex. It sits directly behind your forehead. It is the most recently evolved structure in the human brain. It is the part of you that can hold multiple variables in mind simultaneously, weigh long-term consequences against short-term impulses, inhibit the first response in favor of the better response, understand irony, plan 5 years ahead, feel regret, and recognize that the stranger across the table from you is a full human being with an interior life as complex as your own. It is, in short, the thing that makes you a person rather than an animal. It is also extraordinarily expensive to run. The prefrontal cortex consumes metabolic energy at a rate disproportionate to its volume. It requires constant, stable, high-volume delivery of glucose and oxygen to maintain the complex, sustained firing patterns that underlie what we call conscious thought. It is the biological equivalent of a supercomputer, powerful, precise, transformatively capable, and completely dependent on an uninterrupted power supply. The second brain is the amygdala. It is not behind your forehead. It is deep in the center of your skull, nestled in the medial temporal lobe. It is the size of an almond. The word amygdala comes from the Greek for almond, and it has been running the same basic software in its essential architecture for roughly 200 million years. It does not do irony. It does not model consequences. It does not care about your 5-year plan. The amygdala does one thing. It detects threat. It evaluates the incoming sensory and cognitive stream for any pattern that has historically correlated with danger. Loud sounds, sudden movements, social rejection, unpredictable authority, financial loss, physical pain, the specific facial expression that means someone is angry at you. And when it detects such a pattern, it does not wait for your prefrontal cortex to deliberate. It fires. The amygdala fires before you are consciously aware that anything has happened. The neuroscientist Joseph Ledoux documented this in a series of landmark experiments in the 1980s and 1990s, tracing what he called the low road of fear processing, a direct neural pathway from the thalamus, the brain's sensory relay station, to the amygdala, bypassing the cortex entirely. A threat signal arrives. The amygdala responds. The body begins mobilizing. All of this happens in approximately 12 milliseconds. Your conscious awareness of the threat, traveling the longer cortical high road, arrives somewhere between 100 and 500 milliseconds later. You are afraid before you know what you are afraid of. And once the amygdala fires, it does not simply send a polite notification to the rest of the brain. It sends an alarm. It initiates a full-system emergency protocol that has been refined by millions of years of predator-prey interaction. And that protocol has one overwhelming priority: survive the next 60 seconds. Not think clearly, not make optimal decisions, not maintain the metabolic budget of your prefrontal cortex. Survive the next 60 seconds. And the tool it reaches for first is cortisol. Cortisol is a glucocorticoid hormone synthesized in the adrenal cortex from cholesterol. When the amygdala fires its alarm, it triggers the hypothalamus, the brain's master endocrine regulator, which signals the pituitary gland, which signals the adrenal glands via a hormonal relay system called the HPA axis, the hypothalamic pituitary adrenal axis. The adrenal glands respond by flooding the bloodstream with cortisol. The process takes somewhere between 15 and 30 seconds from initial threat detection to peak systemic cortisol elevation. It is fast. Evolutionarily, it needed to be. Now here is where the engineering gets brutal. Cortisol's primary function is energy mobilization. It breaks down stored glycogen into glucose. It suppresses non-essential metabolic functions, digestion, immune response, tissue repair, to redirect metabolic resources toward the muscles and the survival circuits of the brain. It is essentially performing a forced reallocation of your body's entire energy budget, stripping resources from systems that are not useful in the next 60 seconds and routing them to systems that are. And the prefrontal cortex, that extraordinary, metabolically expensive, slow, deliberate, brilliant prefrontal cortex, is classified by this ancient emergency protocol as non-essential. Read that again. Your most sophisticated cognitive machinery, the thing that makes you capable of language, mathematics, empathy, creativity, long-range planning, and everything else that distinguishes you from a reptile, is classified by your own stress response as a luxury item, something to be powered down when resources are scarce and speed is everything. And cortisol powers it down. The mechanism is not metaphorical. It is not a matter of attention or motivation or cognitive load in the soft psychological sense. It is electrochemical. Cortisol binds to glucocorticoid receptors in the prefrontal cortex, receptors that are, with devastating irony, present in very high density in exactly the regions most critical for working memory and executive function. When cortisol binds to these receptors in sufficient concentration, it suppresses the postsynaptic signaling cascades that sustain the maintained firing of pyramidal neurons in the dorsolateral prefrontal cortex. The neurons are still there. The architecture is intact, but the sustained, coordinated, resonant firing patterns that constitute working memory and deliberate reasoning are disrupted. The supercomputer does not break, its power supply is throttled. And in the exact same moment, the amygdala is not being suppressed, it is being amplified. Norepinephrine, the second major neurochemical actor in the acute stress response, is being released by the locus coeruleus, a small nucleus in the brainstem, and it is doing something to the amygdala that is almost the exact opposite of what cortisol is doing to the prefrontal cortex. It is increasing the amygdala's signal gain. It is making threat detection circuits more sensitive, more reactive, more hair-trigger. It is making you see threats in ambiguous stimuli, hear hostility in neutral tones, read danger into uncertain situations. The prefrontal cortex goes offline. The amygdala goes into overdrive. This is not a metaphor. This is the measured electrophysiology of a stressed human brain. And this is why you went blank. In 1915, A physiologist at Harvard Medical School named Walter Bradford Cannon was trying to understand why cats died in his laboratory. Not from anything he was doing to them deliberately, from fear. Cannon had noticed that cats subjected to intense, sustained psychological stress, the kind produced by the proximity of a barking dog, or by prolonged restraint, or by repeated exposure to unpredictable aversive stimuli, sometimes died. Not from injury, not from any identifiable physiological trauma. They died from what he could only describe as the physiological consequences of the fear state itself. Their hearts gave out, their blood pressure collapsed, their bodies, in some measurable physical sense, simply stopped. Cannon spent years investigating this, and in the process of trying to understand how fear could kill a cat, he became the first person to formally document and describe the physiological architecture of what he called the fight-or-flight response, the integrated whole-body emergency mobilization that the nervous system initiates when it detects mortal threat. What Cannon described in 1915, and what we have been spending the century since refining in molecular detail, is one of the most elegant pieces of biological engineering ever documented. It is a system designed to solve a specific and precisely defined problem. How does a medium-sized mammal survive a sudden encounter with something larger and faster that wants to kill it? The solution that evolution arrived at is this. When threat is detected, abandon all non-essential processing. Maximize oxygen delivery to skeletal muscle. Elevate heart rate and blood pressure. Dilate the pupils. Suppress pain sensitivity. Reroute blood from the digestive system to the limbs, and prime the motor system for explosive physical output. Do all of this in under a second, and keep doing it until the threat is resolved. This is not a bad system. For a mammal navigating a Pleistocene savanna, this is a spectacular system. This is the system that allowed your ancestors to sprint away from ambush predators, to fight when cornered, to survive the kinds of encounters that would have killed a slower responding organism. The fight-or-flight response is not a malfunction. It is one of the most successful pieces of adaptive engineering in the history of complex life. The problem, and it is a problem of breathtaking, almost comical scope, is that it cannot tell the difference between a lion and an email. Let that land. The amygdala, operating on its ancient pattern-matching heuristics, cannot distinguish between the sensory signature of a predator emerging from tall grass and the cognitive signature of a hostile message from someone with authority over your livelihood. Both patterns activate the same threat detection circuitry. Both trigger the same HPA axis cascade. Both flood your system with the same cortisol and noradrenaline cocktail. Both initiate the same forced rerouting of prefrontal cortical resources. The lion requires you to be fast, strong, and reactive. Thinking slowly and carefully about consequences is, in that context, a fatal liability. The email requires you to be precise, nuanced, emotionally calibrated, and capable of holding multiple contextual variables in working memory simultaneously. The fight-or-flight response, in that context, is a catastrophic liability. But your amygdala does not know the difference. It was not designed to. For approximately 200 million years, the things that activated threat detection circuits were things that could physically kill you in the next 60 seconds. The idea that a symbolic representation of social disapproval, arriving via electromagnetic signal, could pose a meaningful threat would be, to any nervous system that evolved before roughly the last 10,000 years, simply incomprehensible. And so, the same machinery fires. Now, I want to take you into the molecular level of what is happening to your prefrontal cortex when this machinery fires, because most explanations stop at cortisol and noradrenaline, and stopping there misses the most interesting part of the story. Your prefrontal cortex operates through a specific pattern of sustained neural activity called persistent firing. Unlike the reflexive, transient firing of sensory neurons, which fire, report a signal, and fall silent, The neurons that underlie working memory and executive function maintain their firing for seconds or even minutes at a stretch. They hold information in an active state, like a juggler keeping multiple balls simultaneously in the air. This persistent firing is the physical substrate of what you experience as holding something in mind. It is the mechanism by which you can remember, mid-conversation, the point you were making before the other person interrupted you. It is the mechanism by which you can keep track of which arguments you've already tried while composing a complex response. This persistent firing is not automatic. It requires active, ongoing neurochemical support. And one of the key modulators of this persistent firing is a neurotransmitter called dopamine, operating specifically through a receptor subtype called the D1 receptor, concentrated in the deep layers of the prefrontal cortex.
Dopamine, binding to D1 receptors in the right concentration, stabilizes persistent firing. It acts like a volume knob dialed to the precise setting that allows sustained, coherent, resonant neural activity to continue without collapsing into noise. Amy Arnston, a neuroscientist at Yale whose work on stress and prefrontal function has been among the most illuminating of the past 3 decades, demonstrated something extraordinary about this system. It operates within an extraordinarily narrow biochemical window. Too little dopamine at the D1 receptor, and persistent firing collapses. The balls drop, working memory fails, executive function degrades.
Too much dopamine, and the same thing happens. The signal drowns in its own amplification. Coherence dissolves. The juggler loses track. Under stress, norepinephrine, the same molecule that is amplifying your amygdala's threat sensitivity, also acts on the prefrontal cortex. But in the prefrontal cortex, at the high concentrations produced by a robust stress response, norepinephrine acts through alpha-1 adrenergic receptors in a way that directly disrupts the D1 receptor-dependent persistent firing. It is as if someone grabbed the volume knob on your working memory and cranked it past the optimal point and kept turning. The signal becomes noise. The juggler drops everything.
This is the physics of going blank. Not a metaphor, the actual electrochemistry. And then there is the calcium question. Cortisol, sustained at elevated levels, as it is during chronic stress or during a prolonged acutely threatening episode, enters the prefrontal cortex and begins doing something that is, in the longer term, structurally more concerning than the acute signaling disruptions. Through its action on glucocorticoid receptors, sustained cortisol elevation promotes the opening of intracellular calcium channels in prefrontal pyramidal neurons. Calcium, in the right concentrations, is a normal and necessary second messenger in neural signaling. But calcium at chronically elevated intracellular concentrations is neurotoxic. It activates proteases and lipases that degrade the structural proteins of the neuron. It can, over time, contribute to the pruning and retraction of the dendritic spines, the tiny protrusions on neurons that form the receiving ends of synaptic connections, in exactly the prefrontal regions most critical for working memory. Bruce McEwen at Rockefeller University spent decades documenting this phenomenon, the measurable structural remodeling of prefrontal cortical architecture under chronic stress, not just a temporary suppression of function, an actual physical reduction in the dendritic complexity of the neurons that constitute your highest cognitive hardware. Chronic stress does not just make you temporarily unable to think clearly, it physically reshapes the instrument through which you think. This is the thermodynamics of a fear state. You are spending your most expensive biological real estate to power a survival response that, in a modern context, almost never encounters the threat it was designed to defeat. And the more often you spend it, the more frequently your amygdala fires the alarm for emails, for deadlines, for social confrontations, for financial uncertainty, the more the architectural cost accumulates. You are, in a very literal structural sense, paying for every unnecessary alarm with a small piece of your cognitive infrastructure. Let that settle.
I want to tell you about a study that changed how I think about intelligence. In 2008,
A research team led by Sonia Lupian at McGill University did something elegant and slightly cruel. They recruited a group of subjects and divided them by performance on standard cognitive assessments, separating high performers from lower performers on working memory and executive function tasks. Then they induced stress in both groups using a well-validated laboratory protocol involving a mock job interview with expressionless evaluators and an arithmetic task performed publicly under time pressure. The protocol reliably and reproducibly spikes cortisol to levels roughly comparable to a moderately threatening real-world experience. Then they retested cognitive performance in both groups. You might predict, intuitively, that the high performers would absorb the stress better, that their superior baseline cognitive architecture would give them resilience, that whatever advantage they had would hold under pressure. The data showed the opposite. The highest performing individuals, those with the greatest baseline working memory capacity and executive function, showed the steepest declines in cognitive performance under stress. Not because they were weaker, because they were more dependent. Their cognitive strategy relied on the full availability of prefrontal resources. They had optimized for the use of sophisticated, metabolically expensive processing. And when cortisol throttled that processing, they had less to fall back on. Their floor was lower than the floor of individuals who had never developed the same degree of prefrontal reliance to begin with. Intelligence, under the right stress conditions, is a liability. This is not a comfortable finding, but it is a finding that makes complete evolutionary sense once you understand the mechanism. The fight-or-flight system was not designed for a world where the highest performers win by sustained, nuanced analysis. The problem we face as modern humans is that we are running an ancient threat response system in an environment where the threats are almost never mortal, almost never solvable by physical speed, and almost always require precisely the kind of sustained prefrontal processing that the stress response shuts down. And we are running this mismatch constantly, chronically, in a low-grade persistent state that our stress response systems were never designed to sustain. Hans Selye, the Hungarian-Canadian endocrinologist who spent most of his career at the University of Montreal and who essentially founded the modern scientific study of stress, described what happens to a biological system subjected to sustained, unresolvable threat in his general adaptation syndrome model. He identified 3 stages. First, the alarm reaction, the immediate fight-or-flight mobilization, the cortisol surge, the adrenal firing. Second, the resistance stage, the body's attempt to adapt to the sustained stressor, maintaining elevated cortisol and norepinephrine levels as the new baseline. Third, and most important, exhaustion. Exhaustion is not just fatigue. In Selye's model, exhaustion is the stage at which the sustained metabolic cost of running a stress response exceeds the body's capacity to maintain it. The adrenal glands literally cannot synthesize glucocorticoids fast enough to keep pace with demand. The prefrontal cortex, subjected to months or years of cortisol-driven structural remodeling, shows measurable reductions in volume that can be detected by brain imaging. The hippocampus, which, like the prefrontal cortex, is dense with glucocorticoid receptors, undergoes measurable neuronal loss. The immune system, suppressed for so long by the stress response's resource reallocation, begins to fail at functions it was designed to maintain. Selye published his foundational papers in the 1930s and 1940s, working from observations of sick rats subjected to noxious stimuli. He identified the physiological syndrome. He named it. He traced its hormonal architecture. What he could not do in 1936 was image a human brain under chronic stress and watch the prefrontal cortex thin. We can do that now. And the images are not abstract. They are not statistical artifacts. Voxel-based morphometry analyses of chronically stressed individuals, caregivers of dementia patients, people in high-conflict work environments, individuals in long-term financial precarity, show measurable, anatomically specific reductions in gray matter volume in the dorsolateral prefrontal cortex and the orbitofrontal cortex, the very structures that make you capable of holding a complex argument in mind, of resisting an impulsive response, of modeling another person's perspective. They are physically smaller in chronically stressed people than they are in normal. This is the physics of modern life. But I do not want to leave you here.
in the anatomy of a brain being slowly reshaped by a mismatch between ancient engineering and a contemporary environment, without also giving you what the research says about the other direction. Because the same plasticity that makes the prefrontal cortex vulnerable to cortisol-driven degradation also makes it responsive to interventions that reverse the damage. And those interventions, when examined at the level of their mechanisms, turn out to be doing something precise and elegant at the level of the biochemistry we have been tracing throughout this entire video. The first intervention is one that almost everyone knows and almost nobody believes is doing what it is actually doing at the molecular level: aerobic exercise. When you perform sustained aerobic exercise, running, cycling, swimming, anything that elevates your heart rate for a continuous period of 20 to 30 minutes, you initiate a cascade of molecular events in your brain that is, in several important respects, the biochemical inverse of the stress response. You increase cerebral blood flow to the prefrontal cortex. You stimulate the release of brain-derived neurotrophic factor, BDNF, a protein that promotes the growth and maintenance of neurons and the formation of new synaptic connections, and that is produced in direct proportion to the duration and intensity of aerobic activity. You suppress the HPA axis response, lowering the baseline cortisol set point, and you promote neurogenesis in the hippocampus, the actual birth of new neurons in the structure that serves as the gateway to long-term memory formation. John Rady at Harvard has spent decades documenting what he calls the miracle grow for the brain effect of aerobic exercise on cognitive architecture. The mechanism is not motivational. It is not about discipline or mood or the runner's high. It is about BDNF and cortisol suppression and hippocampal neurogenesis and prefrontal blood flow. It is engineering. The exercise is doing something specific and measurable to the hardware. The second intervention is more counterintuitive, and it is the one I want to spend the most time on, because it is the one that speaks most directly to the core physics of the problem we have been examining: controlled breathing. I know how that sounds. Compared to the thermodynamics of the HPA axis and the electrochemistry of norepinephrine-mediated D1 receptor disruption, breathe slowly sounds like a platitude. But the mechanism is not a platitude. The mechanism is one of the most elegant bidirectional pathways in the entire autonomic nervous system, and it was not understood at the mechanistic level until surprisingly recently. Here is the architecture. Your autonomic nervous system, the branch of the nervous system that regulates your heart rate, blood pressure, digestion, respiratory rate, and all of the other functions that proceed without conscious direction, is divided into two subsystems that are, in broad terms, antagonistic. The sympathetic nervous system is the accelerator. It is the branch that the fight or flight response runs on. It increases heart rate, elevates blood pressure, dilates airways, suppresses digestion, and drives the cascade of responses we have been discussing throughout this video. The parasympathetic nervous system is the brake. It slows heart rate, lowers blood pressure, promotes digestion, and is associated with the recovery state, what Walter Cannon called rest and digest, the physiological counterpart to fight or flight. Under stress, the sympathetic system is dominant. The parasympathetic system is suppressed. The entire physiological architecture of the stress state is predicated on this imbalance. Now here is the critical anatomical fact. The only organ in your body over which you have simultaneous voluntary and autonomic control is your lungs. Your heart rate, your blood pressure, your cortisol levels. You cannot directly will these to change, but your breath rate you can control consciously, deliberately, and immediately. And your breath rate is directly coupled through the vagus nerve and the baroreflex to your heart rate and your autonomic balance. When you slow your exhalation, specifically and deliberately making your exhalation longer than your inhalation, you increase what is called respiratory sinus arrhythmia, the natural fluctuation of heart rate with the breathing cycle. You stimulate vagal tone. You activate the parasympathetic system's braking effect on the sympathetic cascade, and you begin, within seconds, to shift the autonomic balance that has been driving your cortisol elevation and your prefrontal suppression. This is not relaxation in the vague, subjective sense. This is a direct mechanical intervention in the autonomic nervous system via the one voluntary lever it exposes to conscious control. The research team, led by David Spiegel at Stanford, studying a specific breath control practice involving cyclic sighing, a double inhale through the nose followed by an extended exhale through the mouth, found in a 2023 randomized controlled trial that 5 minutes of this practice daily produced greater sustained improvements in self-reported stress, autonomic measures, and physiological affect regulation than 5 minutes of daily mindfulness meditation, sustained attention, or box breathing. The double inhale appears to be particularly effective because it reinflates the lungs' alveoli, the tiny air sacs where oxygen exchange occurs, more fully than a single breath, maximizing the surface area for gas exchange and producing a more robust activation of the stretch receptors that drive vagal parasympathetic response.
The physics of calming down is not mystical. It is biomechanical. And this brings me to the last point I want to make, and it is the one that ties together everything we have covered. Your brain is not an unchanging machine. It is a dynamic, plastic, metabolically responsive structure that is being continuously reshaped by the demands placed upon it, by the environments you inhabit, the threats you perceive, the recovery time you permit yourself, the physical activity you engage in, the quality and duration of your sleep, and the degree to which you allow your autonomic nervous system to return to parasympathetic baseline between stressors. The prefrontal cortex that goes offline when you go blank is not a fixed piece of hardware. It is a living architecture that is thickening or thinning, branching or pruning in direct response to the biochemical environment you are creating for it every day. Chronic stress creates a biochemical environment that degrades it. Exercise, sleep, controlled breathing, and genuine recovery create a biochemical environment that rebuilds it.
You are, at this moment, either building or dismantling the instrument through which you think, not by how hard you are thinking, by what is in your bloodstream while you are doing it. Put your hand on your sternum. Feel your chest move. Now, slow your next exhale. Make it twice as long as your inhale. Do it again. And understand that what you just did, that simple, mundane, almost embarrassingly easy act of controlling the rhythm of your own breath, has begun shifting the electrochemical environment of your prefrontal cortex. Not metaphorically, mechanically. You are not at the mercy of your stress response. You are in an argument with it. And now, you know the physics.